My name is Mark Farrell. I am the manager of the molecular biology core facility. We're located on the fourth floor of the Barbara Davis Center. I've been the manager for almost 5 years now, and and there's my contact info at the bottom. I'm the only one in the core, so if you have any questions or concerns, um, I'm the person to contact. All right, I'll just jump right into it. So the mission of the molecular biology core facility is to facilitate and promote the application of contemporary molecular biological techniques to existing research programs at the university, its affiliated institutions, and outside entities. Investigators in our research base come from diverse clinical and scientific backgrounds, including the below disciplines. So what does that mean for you? Basically, what can we do for you is I'm sure the question you're asking. To briefly cover a menu of our services, and then I'll go into more depth into each one throughout the presentation. So the first one I'll be addressing is Sanger sequencing. Then I'll be talking about cell line authentication using STR analysis, mycoplasma testing, RNA quality testing with a bioanalyzer, and dynamic glucose-stimulated insulin secretion assay via parafusion. We will also be shortly offering digital PCR. We have the instrument in the lab, and I'm just waiting to be trained on it, so I won't be discussing it today, but that is coming soon as well. So the first service I'll be talking about is Sanger sequencing. Most people know what this is and what it's about, whether or not they need to use it. So I'm just going to discuss it so people are aware of it. Won't get into too deep here. We use a dye terminator sequencing protocol on the ABI 3500, which is pictured right there, that blue and white instrument. We can sequence either plasmid or purified PCR product templates. We are capable of purifying your PCR products for you for an additional cost. We can get reads up to 1,000 base pairs long. We have online sample submission and results retrieval through iLabs, and a convenient Dropbox location in RC1 South on the 8th floor. That Dropbox location can be used for if you're submitting samples for any of our services, basically. We have next day business results for this service. As long as samples are submitted before 11 a.m., which is when I go to that Dropbox to go pick up samples. We have free reruns for samples that meet our sample submission guidelines, which can be found on our website. If you just Google molecular biology core facility CU-Anschutz, it'll pop right up. And we also have common primers available for free through our core facility. And discounts for bulk ordering and primer template premixing. Those bulk discounts apply to a lot of our services, almost all of them. So keep that in mind as well as I move through this presentation. So the bulk of my presentation is going to be about our cell line authentication service, because I think fewer people know what it is, and fewer people know how important it is than some of our other services. So that's what I'm going to be spending the next few slides on. So first of all, when you have cell lines, they can become cross-contaminated or evolve in culture. And you can imagine why that might be bad for your research if you have cells in your dish that aren't the cells you expect them to be. But you can't always tell just by looking at it, right? And when you do have cross-contamination, new cells can outcompete old cells and eventually replace the line that you want to work with. This is actually a really big problem. More cell lines than you may think have been contaminated or misidentified since their inception. And known contaminated or misidentified cell lines are still used in research today, and more often than you may imagine. And I have some statistics on this. So between 1999 and 2015, Christopher Korch, who used to work here on campus, profiled over 1,600 samples and found a number of instances of misidentified and contaminated cell lines. And this data here sort of shows you in the blue you have authentic cell lines, and the red stripe data is misidentified or contaminated cell lines. And you can see that for some of these subgroups, you have upwards of 40 to 50% of the samples that he tested as contaminated or misidentified. And you can also see those last two bars are actually from published data. And I can also say that if you look at the other published data, these numbers are not uncommon or, abstract. So what should you do to make sure that your cell lines are what you want them to be? Before you order your cell lines, you should always check their names on the ICLAC database or Cellosaurus or something similar. Sometimes you can go on these databases and you can find out that the cell line you might want to work with is misidentified or contaminated, and this is already known. You also want to work with, you also want to search for these cell lines on these databases so that you know that there is a reference profile that you can go back to and compare any data that I may generate for you to. So you have a baseline, basically. You should also always obtain cell lines from sources that can demonstrate the authenticity of the material. Some of those places that are more reputable I have listed here. You should also make sure you authenticate your cell lines routinely. This is where we come in. So we recommend that you authenticate your cell lines at the start of your project, throughout the project, whenever you notice a novel phenotype, after you select for phenotypes, so after you select for drug resistance, for example, after every 10 passages, at the end of a project, and before submission of grant applications or manuscripts. And this is also important because more and more journals are actually requiring that your cell lines be authenticated before you publish. And then obviously you should always use best laboratory practices to reduce your chance of cross-contamination. So where do we come in? How can we help you out with this issue? So the logistics of it is you submit an order through iLabs and you give us your pure and as pure as you can get it and accurately quantified human gDNA. We do not test non-human gDNA. We can extract the gDNA for you from a cell pellet for an additional cost. You submit your sample directly to the core or put it in our Dropbox, which I mentioned earlier. We use a global filer kit from ABI to amplify STR sequences at 24 different loci across 5 dyes. For those of you who don't know, STR stands for short tandem repeat. This is a short sequence of DNA that will repeat any number of times. And the locations of some of these STRs are known in the human genome. And this kit is designed to amplify those STRs. And these STRs are also highly polymorphic. So the length of these STRs vary between individuals, and whether or not they are even present on any given chromosome also vary. And so across 20 different loci, 2 chromosomes per sample, you have a large enough data set that's polymorphic enough that we can accurately distinguish between individuals. So after we've used this kit, we use our 3500, which is the instrument that we use for sequencing, to generate raw data of your samples. And then we send that raw data to you in the form of a table and a electropherogram, which you can then compare to these databases that I mentioned earlier, like Cellosaurus. We can do this comparison for an additional cost as well if you'd like. So here's an example report that I might send you. This is control data from one of our more recent runs. You can see that there are these, on the right-hand side, you have the electropherograms, and the gray boxes at the top represent one loci. And the numbers immediately below each of the peaks, the whole numbers with no decimal points, those are that's the length of the STR at that loci. You can see that at each loci you have generally 2 alleles. This is one for each chromosome, right? At some loci, like if you look at TPOX, you might have one one allele, that can either be because the STR is the same length on both chromosomes, or because the STR is only present on one chromosome. And then on the left-hand side, you have a data table, which is the exact same data that you see on the right. It's just in a more readable form. And you can take this data table and go to these databases that I mentioned and pull up this data table side by side with a reference profile and determine whether or not your profile that I generated for you matches well enough with this reference profile. And again, that's the comparison part, and I can do that for you for an additional cost. So the other half of this is Mycoplasma testing. Mycoplasma is a bacteria that can secretly infect and disrupt your cell cultures. And I use the word secretly because Mycoplasma is very difficult to detect. It does not cause changes in media turbidity or pH. It produces few detectable metabolic byproducts, and it cannot be detected under a microscope. And it can easily disrupt your research as well. It can cause chromosomal aberrations, disrupt nucleic acid synthesis, change membrane antigenicity, inhibit cell proliferation and metabolism, decrease transfection rates, change gene expression profiles, and even cause cell death. So it can be highly disruptive. So for that reason, you should perform screening routinely. You should perform Mycoplasma testing when you receive a cell line, after every 10 passages, and when you're preparing a cell bank. Of course, when in doubt as well. And I do this test using gDNA or a cell pellet, just like I would for the cell line authentication service I just discussed. And for that reason, when you combine these two services, I can give you a pretty heavy discount. I cannot test Mycoplasma from supernatant because the cancer center does that. So, but I can use gDNA or cell pellets. Again, here's an example report that I might send you, or part of the important part of the report. For this particular report, you can see that these samples of interest are in lane 14 and 15. 1 through 13 are other customer samples, as well as 16 through 18, and then the last two lanes are controls. So you can see that what we're looking for is 3 distinct bands. When you have 3 distinct bands, that one in the middle is Mycoplasma. And when you only have 2 distinct bands, you know that it's negative for Mycoplasma. You can see that the sample in lane 14 is negative, whereas the sample in lane 15 has a band that's positive. We also offer RNA quality testing. Again, this is kind of like Sanger sequencing. If you know, you kind of know whether or not you need this, but not a lot of people are aware that we do this for you, that we can do this for you. So we have a bioanalyzer that sits right next to me here, 2100 model. Delivers sizing, quantitation, integrity, and purity of RNA samples. It is most commonly used before RNA-seq, and we use a total eukaryote RNA assay. We have nano and pico kits. Those just describe the ranges of concentration that each kit can handle. We do not have mRNA kits. We do not have DNA kits. There's just not enough demand on campus for it. And again, here's a sample report for you. So stuff on the left isn't quite as important, but you can see a gel representation and an electropherogram representation, and then of the RNA purity. And then on the right, you have the RIN scores. This is what most people are looking for. A lot of times when you go to submit your RNA for RNA-seq, they have a minimum RIN score that your RNA needs to be at. So we can generate those scores for you. And the turnaround time for this is a day or two, really quick. The turnaround time for the other 2 services, the cell line authentication and Mycoplasma, that's more like 5 to 6 business days, but I can rush those as well if you need. The final assay I'd like to discuss is our dynamic glucose stimulated insulin secretion assay. via parafusion. This is specific for diabetes research, but we are looking for ways to apply this technique to other kinds of research as well. So if you hear me describe it and you think, wow, that might be interesting for my kind of research, but it's not diabetes, we can have a discussion, we can figure it out. So the purpose of this is to test the functionality of human or mouse islet cells. So the way it works is you have up to 12 different chambers where you put a sample of islets in each one. 20 to 30 islets is more than enough, as well as beads intended to prevent the islets from just running through the chamber when you start passing solutions past them. And then you have one of any 8 different solutions pass through the chamber at any different time. And the nice thing about this is that you can set it up so that you're passing a certain concentration glucose solution for a certain amount of time, and then for 6 of the samples, you can continue passing that same glucose solution past them, and for the other 6 samples, you can pass a different concentration of glucose solution. Once you've passed all these solutions past your islets, we collect the flow through. This is all automated with the instrument we have, with 1 minute of flow represented by 1 well in a 96 well plate. And then you test representative samples from that plate for insulin concentration to determine how the islets from each of the chambers responded to any given solution at any given time. So if that doesn't make sense, here's a sort of graphical representation that I think will clear it up. So you can see that we have 6 samples from this run. For the first 12 minutes or so, we have a low glucose solution running past them, that 2.8 millimolar glucose at the bottom there. And then at minute 12 or 13, we introduced a high glucose solution where you see insulin concentration spike. And then that remains steady for as long as we are passing that high glucose solution through the chambers. And then once we reintroduce a low glucose solution, that insulin secretion begins to drop off until the very end, where we introduce a KCl solution to depolarize the membrane and cause these islets to dump their insulin, basically, as a control to make sure that the islet functionality is working and that the islets are there before another phase of low glucose solution. So this is sort of our standard run that we might do for you. But if you have other ideas of how you might want to customize this assay for your diabetes research or otherwise, I'd love to hear about it. So those are our services. Again, here's my contact info. Mark Farrell, 2 R's, 2 L's with an E in between. The phone number for the lab, the room number is 4207 in the BDC. And again, we have our sample drop box in RC18 on the RC1 South on the 8th floor near the elevators for sequencing, cell line authentication, and mycoplasma testing services.